So I'm up here in a tea house in West Hartford doing a screencast because I should have done this earlier, but I totally forgot about it. Um, so we're going to be talking about cloning once again, but in this case we're going to be discussing not the old version of cloning where you would pick off one of the cells from an embryonic ball of cells, but we're going to be looking at how to clone from an adult, which means that the cells are no longer pluripotent. Instead, they've completely differentiated. So don't forget about those two terms, the concept of pluripotent versus differentiation. So with respect to pluripotent, it means that the cell has not yet committed. So this is what all embryonic cells in the early stages of the development of an embryo are all pluripotent. Whereas the concept of differentiated means that the cell has indeed specialized and will no longer be anything but that specialized cell, whether it be a liver cell, a skin cell, nerve cell, etc. Now, I hope you understand that the thing that makes it interesting here for cloning is if we want to clone an adult, which is something that would be desirable because we would know what the characteristics and behaviors of that particular adult are when we see the adult, then we would want to be able to make a clone from a differentiated cell. So if it's from a differentiated cell, that means it's a cell that is not pluripotent, one that would be committed basically at that point. So somehow what we need to do is take an adult differentiated cell and change it back into one that's pluripotent. All right, my tea just came out, so I'm going to put this on pause for a moment. So this here is really the key element to this whole thing, because we want to be able to differentiate something that is an adult cell. So say, for example, we have a cow, and just like I said in the previous screencast, we know that this cow is able to produce a ton of milk. In fact, let's just say that it produces 4 times the amount of milk as your typical cow. So you would have fewer cows in your barn, and that means less food that you'd have to feed them, etc. So this would be a highly desirable cow, one that, if you could, you'd want to clone so that you would be able to create a herd of cows that produced a ton of milk. So, as we said, if I decided I was just simply going to pick off a cell, let's say from this cow, and it happens to be a skin cell, if I were to then culture that cell, meaning grow it, all I would produce would be a whole bunch of cow skin cells, and that would not necessarily be a very useful thing. We want to be able to clone an exact duplicate of the entire cow. So how do we do that? Well, turns out there's this guy, Ian Wilmot, who happens to be credited with being the first lab, or he was the principal investigator of the lab, to come up with a way to clone organisms from adult cells, from adult differentiated cells. And he was out of a laboratory called the Rosalind Institute. Oops, I just took a look and it turns out it's not Rosalind, it's the Roslin Institute. And the Rosalind Institute is in Scotland. And the Rosalind Institute worked with sheep, and they cloned a sheep from an adult mammary cell in 1996. Now keep in mind that sheep actually had been cloned, but it was the old method that we talked about in the previous screencast. So in this new method with Ian Wilmot, this was a pretty breakthrough technology that occurred. So this is what Wilmot did. He actually had we have to distinguish here. We have something we're going to call Dolly 1. That's the first generation Dolly. And then we're going to call Dolly 2, we're going to call her the clone. Okay? Well, Dolly 1, long ago, before Dolly 2 was actually cloned, had actually been euthanized, and all of her tissues had been sort of set aside in a freezer. And in particular, there was a set of mammary cells that were put into a -80 freezer. And it's those mammary cells, which are adult cells or adult body cells, also known as somatic cells, okay, which is very different from the sex cells. Sex cells are like the sperm and egg. So somatic cells these were, and they were fully differentiated. And if we had just left it at that, I think you understand that if I picked out one of those mammary cells and started to culture it, all you would have are these udder cells. So you'd have a lot of Dolly udder cells. That's it. That really doesn't get you very far. So what Wilmot did is he took one of these mammary cells and he enucleated it. So here's the mammary cell, here's its nucleus, and Wilmot removed that nucleus, chucked it out of there. And this is going to be a very important thing, because don't forget, this is a nucleus that has DNA in it, which is fully differentiated. And then he took from another sheep an egg cell. So let me just sort of scooch over to here a little bit. And this egg cell had its typical, it was a 1N egg cell, right? So it had its complement of DNA that needed to combine with a sperm in order to create the first zygote. But what Wilmot did is he enucleated this egg. Whoops, let's get rid of the whole thing. And left only a whole bunch of factors. Because don't forget, the factors are what's associated with an egg cell. So this went from being a 1N cell to a 0N cell, because it had no DNA in it at all. All it had were those factors. And then Wilmot decided it would be a good idea. I mean, it wasn't like Wilmot was the only one who was doing this, but there were many different labs who were trying to do this, but Wilmot was the one who was successful. He took this differentiated this differentiated nucleus, and tucked it just below the cell membrane of the enucleated egg, and then he zapped both these things with electricity. And what that did is it led to the egg cell incorporating that the differentiated nucleus, like so. And this was a 2N cell, because that nucleus was a 2N cell. Remember, it was taken from the original mammary gland. But the problem is that since that was a cell that was a mammary cell, that means the DNA was fully differentiated, and if they had left it at this, we would have simply had mammary cells that wouldn't have gotten us very far. So this was the sort of revolutionary thing that Wilmot did. He starved the cell. Starved it, which is kind of a funny thing to imagine doing to a cell. But that's the key element to this, because when you starve a cell, it undifferentiates all the DNA. It's that simple. Okay, so now it's not really that simple, but fundamentally, if you withdraw certain nutrients, then it's going to cause all the DNA to essentially turn on. So all the DNA is available for the developing cell. So as a result of having this cell gone through the starvation period, it undifferentiates. Let me spell this out. And it continues to be a 2N cell. But don't forget the very important thing here, which is that inside of this set of undifferentiated set of DNA, we have the factors. So then we take this cell that has been starved and undifferentiated, and we implant it into the uterus of a surrogate mom. And it's very important that obviously this now you could consider to be a quasi-zygote, gets implanted properly, and then is able to develop, and eventually it would divide into many cells inside of the uterus of this surrogate mom, and it would contain the undifferentiated set of DNA, but obviously with the factors that are hanging out inside each one of these cells. Eventually, this would lead to the proper development, and once the sheep came to term and was born, then we had a full-fledged Dolly, and of course this is Dolly 2. It's the genetic clone of Dolly 1. Dolly 1 wasn't alive, unfortunately, at that point, so you couldn't do the skin graft test. That was sort of the way to test for the veracity of your clone. Now, just to sort of show you what's going on here, let me show you a picture of Dolly. Okay, so I have here Dolly. Dolly's the little babe right here. Here's the surrogate mom to Dolly. So this is the baby Dolly. What are they called? What are sheep's? Sheep are lambs, right? There we go, lamb. So we have a lamb, and that lamb was Dolly number 2. And then, obviously, Dolly grew up as a very famous, Dolly's no longer alive, but became very famous. And you can see here, this is kind of fun, the press here checking out Dolly and saying, oh, well, yeah, that's clearly a clone. Of course, they'd had no idea because there was not a Dolly 1 around to compare it to. But there you have it. The key element to this whole thing is that you were able to create a clone from an adult cell so that you could have, in perpetuity, a clone of all those, whatever adult mammal that you might have. Clearly, there are a lot of controversies associated with this. There are some limitations that were discovered, as well as some enhancements that have come about over the years. But the most important, sort of the father of modern cloning, is Ian Wilmot, and there are some people who believe that his work was flawed. In fact, I'm one of those people, significantly flawed, and perhaps we'll have a chance to talk about that in the future. Okay, bye.